Okay, well, thank you very much for inviting me. I obviously didn't behave too badly 30 years ago. So it's a pleasure to come back. I was just doing my PhD at the time when I was working on oysters. I was already beginning to realize I was supposed to do a PhD on the evolution of oysters, fossil oysters. Very, very rapidly during that time, I realized that I actually wasn't that interested in fossils. And I was much more interested in modern molluscs and looking at, if you like, the aspects that drive evolution, but much more interested in, I'm going to say it now, the real thing than fossils, which always have far too much missing for my liking. So that's sort of where I come into this. And as Ben very kindly said, oh gosh, why is that not working? Oh, no, it's not. It won't seem to have gone.
The presentation.
Yeah, that might be.
So when you press the key, it didn't go.
No, is it not in
Maybe
presentation mode?
to do this.
Go back to that one.
Maybe we've got 2 copies of it open. Right, please bear with us. We are screen shares. Disconnected.
So go to that again and share again. So go to the PowerPoint.
Yeah.
Then start the presentation from the beginning. But questions, can they see that?
They can't see it.
No, I don't think they can see
So
that.
if you go back to Zoom and share screen.
Yeah. Share screen.
Yeah.
I can share that, but when I wish, yeah, that's what we were doing, but it wouldn't change slides.
Oh, you will notice it's now in that display.
Yeah.
Yeah.
Back to your presentation.
Back.
Yeah, well, that's fine as it is now. Yeah.
Yeah, but that won't change either.
Well, that's what happens if we do it. Oh, yeah, it's okay.
Yeah.
We'll use that.
Can you see different slides there, please? Anybody at home?
Yep, that's lovely.
Okay, right.
You didn't need to talk.
There
About that. Okay, thank you very much. Well, to add to the chaos. So anyway, that's where I came in. Gosh, I do it in maths, it's worse. So, as Ben very kindly said, my principal interest is always bivalves. And I'm sort of grown into, from looking at oysters and things that live on the surface, I'm sort of expanded into looking at all sorts of bivalves, but actually I do work on gastropods as well sometimes, and there's a couple of pictures of gastropods in here. It's just many of you are snail friends, so please don't go to sleep too much. Many of the things that I say about bivalves actually do map very much across to talking about gastropods as well. I also work on brachiopods, which is a bit of a sort of heinous thing to say. A ontological meeting, but in many respects they are rather similar. So many of the aspects of how you make a shell and how you keep a shell are either the same as molluscs or actually have interesting parallels that we can compare between the groups. But I promise not to show you any brachiopods today. So, one of the things that's deeply fascinating about bivalves is there's just a massive diversification of different life habits and different shapes then that actually are adaptations for those different life habits. This is one of the few groups of people, I think, that I don't have to explain the watering pot shell in the middle and claim that it's a bivalve. So amazing diversity of shape and form. And what I'm principally interested in this respect is actually thinking, well, how do you build those shells? How have you made those great diversities of shape, but also compositions of those shells? So let's just have a look about why you want to do that. I mean, again, I'm preaching to the converted here that that one of the prime functions of a shell is to protect the occupant. And if you like, the threats that you need to protect against are actually rather varied. And therefore the sort of one solution to one threat doesn't fit all. And I've got here just some of the different things that shells have to withstand. Some of them obviously very physical in the environment, being pounded by waves and rocks, and obviously only in the Antarctic at the moment and the poles, in the polar seas, that being crushed by ice, that's a really important threat to shallow water communities in those places. And being crushed by different predators, but also different things like grazing has an effect. You've all seen limpets on beaches which have been well grazed by their friends and other gastropods, all of which are attacking shells. And at the end of the day, if you make a shell, you do it for a reason. It costs a certain amount of energy to produce. So you need to have a shell that you can either keep intact as much as possible, or that you can in some way renew. And the bottom line, particularly for bivalves, rather than almost any other kind of mollusk, is make a hole in a bivalve, even if that's not a successful predation attempt or something, and that is usually the end of that bivalve, either because they leak so many body fluids into the water that it damages themselves, but also attracts secondary scavengers and predators. So the very, very strong evolutionary control, or a very strong selective pressure to building a strong shell that you can keep intact. It's no good laying all that energy down and out for nothing. So that's sort of where I'm sort of coming into this. And of course, to one of the things we're trying to think about is how the modern oceans are changing, but also looking back over geological sort of timescales. We know that oceans have changed a lot, both in the non-biological parameters, things like temperature, salinity, pH, but also the biological pressures have changed in terms of competition, and also predation pressure has changed massively over the last 400, 500 million years. And of course, again, on human timescale, we are disturbing environments. So these oceans are changing over various, if you like, length scales. What effect does that have on the molluscs and how they build their shells? And that's one of the things that I've been very interested with my students over the last few years, is actually looking at familiar shellfish, like mussels in this case, but we've also looked at dog rocks and things like that, and actually gone back and looked at historic collections which have been collected at different times in the last 100 or so years, to see if basic shell parameters, things like thickness and what they make their shell of, has changed during that time. But also, in a slight nod to my past, actually going back with some of those species back into deeper time into the fossil record. So here we have a bunch of essentially markerless edgeless, from ones obviously living on the shore now to ones from the red crag in Suffolk, which are perhaps a couple of million years old. And we can look at those shells to see, same species, are they changing in thickness and what have you over time. So just to recap, I think probably all of you have an idea about how molluscs make their shell, but just to sort of recap, that's the same thing for the gastropods and the shell of cephalopods, doing the same thing. So then we've got our jolly bivalve at the top, and I'm just going to cut it along that midline, along the sort of maximum growth direction. And you know that the shell is underlain by this very thin tissue, the mantle, and that mantle is responsible for laying down the shell material. Okay, and the very first part of shell material it lays down is what's called the periostracum, this organic sheet. You're very familiar with seeing that on things like mussels that have a very thick organic sheet on the outside. It's secreted by cells at the edge of that mantle, but then it reflects back over the shell. And if you like, that then forms the template onto which the rest of the shell, the calcareous part of the shell, can be laid down. It's that organic sheet that actually controls the shape of the shell, and things like mussels that have a very thick organic template can't really do anything very wacky in terms of the outside morphology of the shell because they can't bend that sheet. That sheet might be several hundred microns thick. You can't bend. It's like trying to do origami with very thick cardboard. Whereas bivalves like scallops, for example, spondylus, oysters have very, very thin organic sheets, maybe a matter of sort of tenths of a micron. And with that, you can build a very intricately shaped shell. Okay, so that's one of the sort of controls that we've been looking at. But the key thing here is to remember that the shell is principally going off to my left. Okay, it's growing in that direction. But all the while, as these growth lines here are trying to show you on this strange cartoon, is that the mantle is also thickening the shell. So bivalves, brachiopods, all of these groups with nice accretionary skeletons show you the shape as they grow over time. And we can sort of, if you like, pull them apart chronologically. So that's what the shell is like at one level. So let's now go zoom in, if you like, onto the, I'm sure it's a mirror image now. Zoom in right at that very edge of that shell. Okay, and there's the periostracum again, the brown layer coming out, reflecting back, and the shell just underlain by the mantle, getting thicker and thicker as you go again towards the right as you look at it. Okay, and all shells have that same basic architecture. Organic sheet, the periostracum on the outside, often doesn't even last the lifetime of the mollusk, let alone afterwards. And then in this case, you can see that the calcareous part of the shell is laid down in layers on top of that. And this particular cartoon has got two different shell layers. And what this SEM over here on the right is trying to show you, it's a Chambardia, one of the freshwater bivalves. Try to pick that one because it has a nice thick periostracum. So you can see fracture through the shell. You can see the periostracum at the top. And then you can see one shell layer, which is presenting these rather beautiful coarse prisms. And then a second shell layer underneath that is a form of nacre. Okay, and that's what all shells look like. All shells have more than one shell microstructure, and they build, if you like, sequentially inwards. So what's that shell made of? Okay, well, it's made largely of calcium carbonate. Okay, and the thing you should know about calcium carbonate is that it has several different polymorphs, several different types. So the chemical formula is the same, but the atomic structure is different. Okay, and as far as we're concerned, looking at molluscs, there are two very important ones to think about. One is calcite, which is a name that you're probably most familiar with. Another is aragonite, which is actually the principal calcium carbonate that molluscs use. Okay, that's a slightly strange thing that molluscs do favour aragonite, because actually in the real world, away from biology, aragonite is actually only stable in sort of very low-grade metamorphic rocks, so high temperature, high pressure. So it's very strange that things like the molluscs, like the modern corals, etc., actually use that as the calcium carbonate of preference. Okay, I've just been up in the mineral galleries. It was very sad, beautiful minerals to be seen. And nobody was looking at the calcite or the aragonite. Everybody was looking at all the other more beautiful minerals, I suppose. So that's the basic sort of slate of crystals that we need to, is it right?
It's fine, we're just trying to work out what we can do. No. There.
Let me see what you mean. I could have done that longer. Tell me next time and I'll do it. So those are the basic carbonate minerals, but I'm very, very pleased. Somebody came in, so I don't know your name, but there is some exception to that. So the thing over on the left is corbicula. I hope that's the right name. I'm learning lots about how many species there might be. That's a very standard happy corbicula. Corbicula only lays down a shell of aragonite. It uses a structure called prostamella, that doesn't matter, but nothing exotic like nacre. And your average happy corbicula looks like that. Look at these ones on the right. Okay, now we came across these about sort of 10, 15 years ago. David Aldridge, that some of you may know in the Cambridge Zoology department, who's very interested in freshwater bivalves, had a student who came across the road to sit with me to see, to show me these ones here. And we spent a very unhappy day looking at those shells, trying for me to make them fit what I thought I knew about bivalves. And in the end, I gave up. Well, I didn't give up. I pushed the matter on and actually crunched up this strange green stuff and sent it off to XRD. And it actually turns out to be an even more rare carbonate mineral, a thing called vaterite, which is really very, very unstable. But somehow it's associated with these corbicula, obviously pathologically. Okay, so I don't know how well you can sort of see that, but the vaterite, the strange mineral, is these big bulges. Those big bulges sort of actually form into the valve. They distort, or they remove space from where the animal's living. It clearly isn't a very good idea for them. They change the shape of the shell, which is one of the things that I'm interested in, one of yours over there, because they get so they can't shut properly. They change their general shape. So it's one of I thought to put this slide in just because I know that members of the Conc Stock are actually very good at finding things and recording things. And actually, I think we would very, very dearly like to know how many populations of corbicula have this strange mineralization. So the original ones that David got were from the River Yare in the Broads, but we have since discovered that the ones at Richmond Lock are absolutely peppered with this stuff. And it looks like they never recover from it. Okay, so it looks at pathology, and I think probably in the end kills them. But it just shows you really that, you know, people have been looking at shell microstructure and mineralogy for 200 years, and yet there is always something new to learn. So let's have a look at, we've established it's calcium carbonate. Let's have a look at the form of that. Okay, so shell is a composite material. It is principally made up of a sort of stiff.
Okay. No, I can't. Yeah, thank you.
So shells have a stiff crystalline component, which is the calcium carbonate. And that's hugely diverse, the forms that different molluscs use. So there's no need to look at all of these and worry about what the name of the species is, or indeed worry about what those microstructures are. But I've just put this in really, just so you can see, look at the scale bars. Okay, they vary between sort of 50 microns on one furthest left to a couple of microns for some of them. Look at the scale bar, look at the shapes of those crystal units. These are really very, very diverse microstructures that different mollusk groups have picked up. And the interesting thing is that some of these are calcite, some of them are aragonite. You never swap. So this, for example, here, the second on the right from on the bottom row is bog standard nacre. That is always aragonite. You can't change that pattern. Okay, so it's very fixed. And the other thing that we know in mollusk shells is that each shell is made of more than one microstructure, and again that is very fixed for that particular species. Okay, so there we have the crystalline component. Why, actually rather beautiful to look at on a scanning electron microscope. I spend a lot of my time doing that, but also just a fascinating sort of hierarchy of different shapes and sizes. It's not just crystalline. It'd be a very stupid idea to make a shell that was pure crystal. It would fracture and cleave very easily. So all of these shell microstructures have organic material within the shell. And this over here is a visol, it's a perioid. And what I'm looking at here is onto the outside of the shell with the SEM, and I have deliberately over etched it with acid. So that acid has eaten away some of the calcium carbonate crystals. Calcium carbonate crystals in this case are sort of nice long prisms, and you can see that each of those prisms is surrounded by an organic jacket. Okay. And we know that those organic jackets around the crystals of the shell perform a function. Okay, so shells with sort of high levels of these organic jackets tend to be really very flexible, or at least while they're alive. Okay, they tend to become very brittle when the animal's dead and it's dried out. And it also helps actually prevent cracks propagating through the shell. So it's really very important mechanical part of the shell. But the other thing we think is we think it's very, very to lay down organic, rich microstructures. So that's an interesting thing that I haven't sort of prepared a slide on this, but the strongest and toughest molluscan microstructure is nacre, okay, mother of pearl. It's the one that they all lose evolutionarily. Okay, so actually relatively few molluscan groups have kept that primitive microstructure, but it is by far the strongest, which is a sort of slightly interesting concept. Why do you do that? But the other thing about nacre is it has a very large organic component. And it may very well be that they're just dropping it because it's very expensive, and you can do as well. You can make, rather than make a strong structure, you can make a strong shell just by simply making the shell thicker. So the amount of organics in the shell varies massively. Okay, so one of the things I do is measure the amount of organic material within the shell. Some species, like the Osogramon I've just shown you, that is a very high percentage. It's 12% of the dry weight of a shell. That's a huge organic investment for the animal to lay down. Whereas this is calmer. I don't know how many of you are familiar with those, a jewel box clam. The calcite on the outside of that has almost no organics in it at all in the outer shell layer. And actually, when I look at these crystals, you can see they're not so beautifully constrained as in Isogramon. And actually, when I look at these crystals, in many ways, they are very similar to geological calcite rather than biogenic calcite. So we know that those organics do a lot, okay, in controlling the shell microstructure, but we know that there's an advantage to them in that they make the shell more flexible, tougher, but that they probably have a cost. And it's a balance between those two things that the molluscs are doing. So what do we know about the very early molluscs? I mean, I've just alluded to the fact that they were all nacreous, but what do we know about them? Well, this is the very earliest bivalve, thing called Podgetaia, that appears in the Cambrian. They are very small. I always say nice people don't look at this kind of thing. They're very, very small, and they're preserved entirely as internal molds. So there's no shell there at all. And that again is a disadvantage for the geologist rather than the animal of laying down aragonite. Aragonite is not stable over really any long periods of time at all. So you tend to lose it. So these tiny little Cambrian bivalves, we don't see the shell, we just see the internal mold. But these are pictures from my colleagues in Spain who are able to look at the, if you like, the outside of this mold, and they can see the pattern of the crystals that were in the shell. And so we know a bit what these were made of. They were made of aragonite, and they were made of sort of plates very much like what you would see in modern nacre. Okay, so primitively all molluscs are aragonite. Okay, cephalopods, gastropods, bivalves all made their shells, albeit out of different microstructures, only using this particular polymorph calcium carbonate called aragonite, which, as I say, is not a geologist's friend. But one of the things that sort of interested me, and what I'm going to try and talk to you a bit about today, is that repeatedly the molluscs have, making it sound very anthropomorphic, sorry, but repeatedly decided to add calcite to their shell. So aragonite is the primitive structure, but they have repeatedly picked up the ability to produce calcite. And again, totally separately. Okay, so it's not a single group that suddenly found calcite and they've exploded evolutionarily. It's different groups that have picked up this ability. And we can sort of see that even across the different classes. Okay, so bearing in mind again that everything starts off with aragonite. There are many of the gastropods, many of the limpets. These are some dog rocks. They have calcitic shells, or the outside layer of shell. The bivalves, here are the oysters. They are almost entirely calcitic. But even the cephalopods, bless their heart, you get used to sort of thinking about nautilus with its pearly, beautiful aragonitic shell. But even within the cephalopods, there have been several entirely independent attempts to use calcite, from these beautiful belemnites, from the squid-like cephalopods to the aptychi. That's a really interesting thing. Ammonites entirely used aragonite to make their shell, but the aptychi, the things which are either jaws or opercular, depending on who you are, those are made of calcite. And even something that Antonio Checo and I are very interested in at the moment are those egg cases of the Argonaut octopus. Somewhat bizarrely, they're a bit bizarre anyway, but they make them out of calcite rather than aragonite. So that's something that's always really interested me. Why do molluscs go from being aragonite? What are the selection pressures? What are the advantages to suddenly picking up calcite on your shell when you started off making a shell out of aragonite? And it's actually quite a difficult thing to solve. Oops, let's move this one. So, oops, I don't need anybody to get too excited or anything about this. This is a large scale molecular phylogeny, or molecular and morphological phylogeny of the bivalves, okay? And I'm not expecting anybody to read any of the branches or get excited about it in any way, shape or form. But that's basically the sweep across the bivalves, okay, from a paper we published about 10 years ago. What I've done is put in yellow the ones that use calcite within the bivalves, and of the whatever it is, 100 odd taxa that we built into this tree, essentially there are two big groups. Okay, they're all within the pteriomorphs, so that big group that has the scallops, oysters, etc., and mussels in it. What I can, however, also tell you is that there is one little group, thing called Carmen the jewel box clam that I mentioned a few minutes ago. One, or sort of very small little group of species within that family appear to put down calcite. It just happens that we didn't have one of those species on this tree. During the Mesozoic, there were these very big, bizarre bivalves called rudists that dominate the sort of Tethyan shelf carbonates. In fact, they're taking the role really of corals before corals come along in sort of reef-like forming things. And they are, again, almost entirely made out of calcite, but they don't appear on this tree because they're extinct. Okay, so we can't plot them in, but they belong somewhere over here. Okay, and the other thing that's wrong with my tree here is that I've got two big groups of calcite secretors. But that's really because I included Mycalus edulis, which is in that furthest left yellow box, and there's a picture of it on the top. That was the only mussel we had in this phylogeny, and Mycalus edulis itself has calcite on the outside. I'll show you in a few minutes. But actually, there's a whole bunch of other mussels that don't. So it's a really difficult sort of game to play with, because actually it seems in general calcite typifies really quite large groups, and so it's very difficult therefore to think about looking at some that do and some that don't and try and tease apart why that might be. And the sort of greatest hope for me has always laid with the mussels, because as I say, some of them do and some of them don't. What's happening now? Okay, so it's a really useful phylogenetic character. If you like building trees, we discovered that shell microstructure was better than many of the other morphological or anatomical things that you might look at in a bivalve. But I'm much more interested in why they do it. Okay, so does it link with specific environments, and why are they doing it? Are they doing it because they can't help it, or are they doing it because it's specifically good for them in some particular way? I'll come back over some of these points in a minute, but the interesting thing in both the bivalves and the gastropods is however many times calcite's around, it is always in the epifaunal ones, the ones that live on the surface, never on the ones that actually burrow. But it's really difficult to pull to pieces, because actually, having said, oh, here's a bunch of bivalves and they all put calcite on the outside of the shell, they're doing it in different ways. They use different microstructures. So it's very difficult to sort of tease apart, sort of say they really all, it's all the same, because actually they clearly aren't all the same. And it looks very much as those separate groups are evolving it independently, and they're doing it in their own way. So there's an advantage perhaps, or a physiological necessity to lay down calcite, and you just solve that problem in your own individual way. Okay. So aside from the mussels, it's just common across whole large groups. And for a long while, I have been desperate for somebody to produce a molecular phylogeny of the mussels so that we could actually map onto that where the calcite was and where it wasn't. Okay. But just to sort of, reiterate, very much has always been the idea that the mussels are primitively aragonitic, and there are some groups that have picked up calcite. Okay, so let's just have a look at, are there any advantages, or what's the pattern that we can see? And I've just sort of said that. So all calcitic molluscs, whether it be bivalves or gastropods, are epiformal. It is predominant in the intertidal or fairly shallow subtidal ones. Okay, it tends to be those guys that hang around the shore. Having said that, the deep sea vent mussels also use calcite, so it's not universally true, but it is very much an intertidal, shallow subtidal thing. It's always on the outside of the shell. Okay, you never put calcite on, at least for the first time, inside the shell. It goes on the outside, but sometimes it spreads, if you like, evolutionarily inwards. I said that oysters don't tend to use anything but calcite, okay, but they came from groups where the calcite just went on the outside. And over time, that layer has thickened, okay, and it appeared, anyway, when I put the slide together, that calcite was always added. Okay, it wasn't transforming, it was always added to the shell. And you can sort of play all sorts of games, get to your geological textbook and say, okay, what's the difference between aragonite and calcite? And you find that aragonite is slightly harder, but not much. And you find this picture over here, this is calcite. Calcite cleaves. If you whack a calcite crystal, it breaks very easily. Aragonite cleaves, breaks less easily. So put like that doesn't seem like a brilliant thing to put on the outside of your shell. There's a very slight difference in the density, but it is very, very slight, that you can sort of pick up in reagent grade crystals rather than the individual microstructures. What's always been attractive to some of us is that there is a difference in the solubility of these two minerals. So calcite is a reagent grade, so again, not in the shell, but reagent grade, just crystals, is 35% less soluble than aragonite. And that sounds really attractive, okay? And I would love to believe that that's what drives this calcite, okay? So we know that dissolution is a big problem. I sort of mentioned this at the beginning of the talk. If you're going to go to the trouble of laying down a shell, you don't want it to start to dissolve, okay? So this thing up on the top left, a Equiodia eitzii, is one of the Antarctic bivalves. It's you can see around the umbo, it's horribly corroded, okay. But the other thing you can see is something that you can see down here on, I don't know if it is Julio actually, there's this freshwater bivalve down here, and also the Bapimoriolus, the deep sea vent bivalve. They're all corroding really quite fast, but only when their periostracum is gone. Okay, so that organic shell layer on the outside almost trumps any other kind of adaptation they have. As long as it's there, then they're protected from dissolution. The minute it starts to get lost, then the shell actually dissolves really very, very quickly. Okay, and I've got some more pictures later of the pteropods. But the interesting thing is these are all animals that live in environments where there are high dissolution rates. And yet many of them have never thought to have calcite. So again, it makes me feel slightly uncomfortable about it. Perhaps calcite isn't the killer solution to being dissolved. So let's go back to those mussels, because I said that was something that I've always wanted to do. So look, here are two mussels that you probably are familiar with. One of them on the left is Mytilus edulis, our blue mussel. That has a calcite outer shell layer. And looking at the inside, I always tell my students when they're going to measure these things, it's kind of handily color-coded, yeah? So the blue layer on the outside of the shell is the calcite, the white layer inside is the mother of pearl. This is Perna viridis, okay, the green lip mussel, and that has no calcite. It is entirely aragonitic. And again, the general idea that we all had without thinking about it was that this was normal and that was not, okay, that this was some exciting extra thing that had happened in the blue mussels and others. Let's just look at those blue mussels a little bit more, okay. So here's the same mussel, set in resin. I like setting things in resin and then cutting it along that XY line and polishing it. And you can see that that blue calcite layer is on the outside of the shell. The white stuff is laid on the inside within the paleoline, and it just gets thicker and thicker as the animal gets older. That calcite has a really, really distinctive structure that's very, very different from any of the other calcitic bivalves. And it's what we call anvil-headed prisms. So if I bleach the inside of the calcite, just remove an organic layer, you can see that it has very, very distinctive morphological pattern. It doesn't look anything like the calcite in any of the other molluscan groups. So it's pretty special. It's got a certain amount of organics in it, but it's very, very distinctive. And I sort of said, well, we always want some molecular phylogeny, and in a way we sort of missed the boat. There was a time when people were producing molecular phylogeny left, right and centre, and somehow bizarrely the mussels sort of got left out. But in about the last 18 months, a molecular phylogeny has appeared. Okay, and again, you don't need to read that in enormous detail. Produced by Koshang Tan in Singapore. And just a really beautiful study of looking at some new mussels, but also taking molecular sequences from elsewhere. And he has produced, if you like, the very first backbone shot of how the different mussel groups relate to one another. Okay, some of which I think we thought we knew, but some of it's been a bit of an eye-opener to some of it. So this came out, and I have had a very keen undergraduate last summer. He didn't know what he wanted to do, but he thought he might like to play with shells. So I persuaded him what he would actually like to do is cut them up and crush them up, which I think probably wasn't what he wanted. He's a zoology student, I think he wanted to love them, but I had him be more destructive than that. But he basically said, look, we've got a molecular tree here. If I can find you one of all these sampled taxa, you can sit there on the SEM, which he looks as though he's enjoying it, and you can crush it up and you can do XRD, you can learn all these things, and we'll find out where the calcite is on this tree. And as I say, I was expecting it to be the odd little terminal twig, okay, but actually, this is a Mars tree. I haven't fiddled with it, so it's missed the species. I've just got the genera, but that doesn't matter. And again, the details don't matter at all to you, okay? But the blue is where the calcite is, and the red is where the aragonite is. And what I really want to encourage you to see is that the mussels, according to these trees, have this very, very deep split in them. Okay, so Michaelis, Perna, etc., are on one branch, and things like Modiolus that we're very used to seeing are on the other branch. Okay, and that split, depending on how the paleontologists feel, that split is of those sorts of orders. So at the very least, sort of 170 plus million years ago, maybe rather more than that. Okay, but what's really sort of striking from what Amal found was not just that the calcite was in both clades, but this really, really distinctive microstructure was in both of those branches. Okay, and you can kind of think what you like about that, but I think that that is so distinctive. I cannot believe that they have been evolved twice in a very closely related groups, which sort of starts to imply actually that the calcite in the mussels is the primitive form, and that actually somewhat it's reversed the question in our minds. So that things like Perna, the green lip mussel, and Septifer and Brachydontes, there's something about those which is making them stop laying down calcite. So it sort of changed the evolutionary question in our minds. I've got time to go a little bit long. Yeah, okay. So I'm now just going to show you another couple, just sort of think about thickness of shell. Okay, having laying down a shell, as I said, is important, but it costs energy. This is another one of my students. He's since gone on, but he had a project just to look at Michaelis edulis or Michaelis galloprovincialis. From lots of sites in sort of Western Europe and up into Greenland. And he did lots of different things. We were interested in how the shell was different at different latitudes. But this is a sort of interesting thing. This sort of particular results, I think, are quite interesting. Look at the graph, don't get too excited about it, but it's plotting the latitude that the mussel community comes from, and it's plotting the thickness of the shell. Okay. And again, you don't worry about the prismatic and the nacreous, although they're both doing the same thing. But just look at the top line, the black line, the whole shell is. As you get into colder and colder waters at high latitude, they are laying down thinner and thinner shells. And they have a bit of a problem because there's two different possibilities explaining that. One is that it's difficult to lay down a shell at low temperature, and we know that generally speaking that is true. So maybe that's what's making the mussels thinner at high latitude. But the other thing is, goes back to, well, why do you have a shell? And at higher latitude, predation pressure is much obviously less, okay? So in which case, perhaps it's a decrease in predation pressure that actually allows them not to invest so much energy in their shells, okay? And similarly, again, I don't want to bore people, but I'm just going to show you this bit. If he looked at the amount of organic there was in that shell layer, and again, remember earlier I said that organics were expensive, then sort of rather interesting difference emerges. So he's got two sites here, northern Norway and Greenland, and again, the details don't matter. This is sort of the northern latitude sites. These are the sites around the UK, and looking at the weight percent organics in the shell. And that surprised us rather, because it looked like at higher latitude, there's more organics going into the shell, which would actually make it more expensive to lay down. And that's either because actually it's more difficult to pump the calcium carbonate around in cold temperatures, or maybe there's actually a benefit to the organics. And one of the things we know, and really both those things are related to the same thing, calcium carbonate is odd. When you want to dissolve the sugar in your tea, you heat the water up. Okay, calcium carbonate is a lot more soluble in cold water than it is warm. Okay, so both of these things sort of both work in the same direction. It's difficult to persuade calcium carbonate to come out of the seawater to make a shell in cold water, but also that shell, when you've got it, may start to dissolve quite rapidly. Remember I showed you the polar bivalve that had lost some of its periostracum. And so we wonder whether actually the investment of organics is actually worth it because it covers the crystals and helps save them. So just while we're thinking, promise I'll stop soon, about changing oceans, and I did promise people the odd gastropod. These are pteropods, okay, and these have always been the poster child of worries about ocean acidification. Pteropods, as you know, are really important in food chains. And there's your Yoldi on top, just to remind you how fast dissolution might go. This is a pteropod shell that I put on the SEM. That's a 30 micron bar. Those beautiful clear shells are only a matter of microns thick. Okay, so it's not a great surprise that people are worried about, well, what happens when they start to dissolve. Look at these funny lumps on the outside. Nobody's ever looked at those. They're the spikes on the side, outside of pteropod shells. But actually, when you get down to it, they're not doing so badly, the pteropods. So again, with the Antarctic survey, there's always been lots of sort of, I'm going to say hysteria, and that might not be the thing to say, so lots of excitement, you know, these things are going to dissolve and it's going to be awful, it's going to be awful. But actually, when you look at them, when they have been caught alive, then actually there is very little sign of dissolution on the outside of them. And the places where you get dissolution are seen in this top right-hand one. You can see some loss of shell material there. That's where the periostracum has been removed. Okay, so if you like, that's an accident that's happened to that pteropod, maybe grappling with other pteropod predators or what have you. If that periostracum goes, then the shell does start to dissolve. But as long as they can keep that periostracum intact, it's like weighing a cagoule in the rain. They're fine. One of the reasons people get over upset and excited about it is that they look at dead pteropods in sediment traps. Of course, when they're sitting in a sediment trap and the tissue has rotted, then the water has gone inside the shell as well as outside, and they start to dissolve very quickly then. But that's fine. Pteropod doesn't care. We care, but pteropod doesn't care. And this one on the left hand is looking with a CT, trying to look at the thickness of, I think, in part this shell here. Okay. And again, you can see actually there are areas of the shell. So the shell is getting thicker. The more yellow the colour, the thicker the shell is. What we can see there is that the pteropods are actually repairing their shell underneath the areas that start to breach. Okay, and that's something we're seeing in the mussels and dare I say the brachiopods as well, that actually they can compensate the shell loss on the outside. I really don't know how they know where the shell has been lost. It's difficult to understand how they can sense that, but they do. So on the one hand, that's really very good news. We can acidify the ocean. These things are going to survive, but it's going to cost them. Okay, and anything, as I keep saying in this talk, calcifying is very, very expensive metabolically. If you've got to keep laying down new material, that is energy that you're not spending growing. It's energy that you're not spending reproducing. So my slightly gung-ho, it's all right, is probably wrong. Yeah. So they are having to compensate for shell loss. So I was going to call it to say that, but I was just changing the subject. Just at the end. These are glass scallops, okay? Deep sea scallops, beautiful things. I mean, you can see why they're called glass scallops. You can see right through that shell. So that's a living one. It's come, from really deep down in the trenches. Amazing things. Very, very thin shell, calcite on the outside, aragonite on the inside. Can you just about see it's got some funny little points? You can see the growth lines, but you can see some funny little points on it, okay? If you look at them on the SEM, we've false coloured this one. That's what those little points are. They grow these most extraordinary things like television aerials off them. And we're just deeply fascinated at the moment to know why would you do that? Yeah, they really are extraordinary. So again, that scale bar at the top is 30 microns. These are tiny. You can see why things like spongus have big spines, but these are really rather amazing structures. So I was just going to leave you with that. So thank you very much. I know I've rambled on, but thank you. Thank you.
Thank you very much, Liz. That was outstanding. I'll just stop sharing there. Get my cursor back on this screen. Oh, this is really difficult to get through. There we go. Right. Thank you very much. Since I'm standing up, do we have any questions from the audience at home, first of all? No, not at the moment. Anybody here? No?
Shocked you all.
I've got a question.
Go on then.
Yeah, Peter?
When a shell gets a foreign object in it, a bivalve, and it overcompensates that foreign object by basically the classic things making a pearl.
Yes.
Or you just have a very, very thick area. That's presumably mostly is nacre.
Only in things
Right.
that make nacre. And as I say, actually relatively few things do make nacre, but things like the the terry pearl oysters, they are doing it with nacre. But some oysters produce pearls that oysters don't have nacre. So they're just using their normal shell material to do that.
Whatever that is.
Yeah, whatever that is, yeah.
Okay.
And I guess pearls always look rather beautiful. So although so the freshwater pearls are also nacreous. So yeah, there's some an addition to that. People think about those pearls because they look rather beautiful and iridescent, and nobody gets excited about oyster pearls or.
So I found mice in this edge of this one or two where a lot of the inside of the shell is taken up by these great big growths inside, which obviously cover it.
Well, it's often things like sponges coming in as well. So when the shell has got lots of endoliths, lots of growing organisms in, they're doing the same thing. They're kind of somehow, I don't know how, but sensing the shell is getting thinner above me, best to box that in.
Yeah.
But also, I mean, some of the Antarctic, there's a thing called Latinula, that's quite a deep in the Antarctic. Just occasionally, they seem to, it must be storm events or something, they get massive incursions of sand, gets down into the shell, which they then just politely sort of brick into their shell. But so several millimeters thick, almost entirely sand layer within the shell, but they identify as alien and just took it out.
Why do you make a pearl? Is that to get rid of a bit of gravel or?
Yeah, so it depends, so pearl gets used in sort of different sorts of ways, but it's really just a sort of, if it's a sort of freestanding sort of spherical drop-like thing, it's usually either a parasite or a sand grain that's got into the mantle, and you know, it's an irritation, and you just smooth
And you
it
just
over
cover
and
it,
make it nice,
cover it up.
yeah, just nice, yeah.
You clearly get pearls around parasites, don't you?
Yeah, so pearls and sand grains is and of course that's how people produce
That's
artificial
another one,
pearls,
isn't it?
yeah.
Do we have possibly non-marine question from John Hutchinson there?
Well, no, not non-marine, but I wonder if there are any evolutionary trends in shell structure associated with the transition between marine, brackish, and freshwater.
No, alas not. So the groups that have gone into freshwater have very different sort of shell structures. None of them have thought about having calcite. But one of the sort of key points that you see in all the freshwater bivalves, regardless of where they come from, and also the gastropods too, is that they are groups which have very thick organic periostrata. So again, it goes back to my sort of, you know, that's the killer adaptation if you want to live somewhere corrosive, is that you have a thick periostracum, but once it's gone, you can't repair it.
Okay.
But yes, things like the corbicula, they don't have nacre in their shells. Things like unionids do. Tricenas, they have a rather similar shell structure to corbicula.
Any
Think
more
that-
questions from there? Oh, I've got loads and loads of questions, but that might be because I didn't understand anything. But
So that's my fault.
with things like rudists, then,
Yes.
and does any of this help you work out that a rudist is A, a bivalve, and B, what kind of bivalve it was closest to?
That's an interesting
I
question.
didn't understand that.
So rudists, you can tell they're bivalves when you look at them. I mean, they've got hinges, they've got muscle skulls.
They look like
Well, they just
a lava lamp.
well, yes, they just have, you know, so oysters have often have a very inflated left lower inverted commas valve. Rudists have just done that in spades. You know, they just have a valve that becomes like a cone, and the other valve is like a silly little flat bit. But yeah, they have hinges and they have adaptive muscles and things like that. Unfortunately, it comes into that big sort of group, the impodents on the end of the bivalves, and they're actually quite conservative in their shell microstructure. So it's not very easy to plonk the rudists where they belong. People liked at one stage to think that they were related to the jewel-bocker clams. Other people thought they were related to things like cardites, but it's very difficult because in a way they're so aberrant with relatively few characters. It's difficult to place them, but somewhere over there on the tree. But yeah, they're bivalves, promise.
Can I ask another question? So these surfaces within prisms and things are very, very tiny.
Yes.
So it would be hard for something like a starfish to feel the difference. They're too small for that, probably.
Yeah, I don't.
But they might, in terms of fouling and things that lived on them, colonized them, could that communicate something to the predators? And that might be one way of hiding yourself by covering yourself.
Well,
calcite,
certainly, yeah. So I don't know whether the microstructure or the mineralogy does that, but I mean that's certainly what things like Spondylus and Karma do. Is you know their spines there are not really so much defensive as in, you know, like a hedgehog or something.
Right.
It's actually attracting epibionts to come and live on them, and then all the things which are smelling and sensing you kind of get lost in the mess that you've got growing on you. But yeah, whether that's those funny aerials in the deep sea or something like that, I don't know. They break very easily.
They look like stinging nettle.
They do. It took quite a lot of preparation because the stuff out the deep sea has lots of sort of very, very fine-grained crud on it that doesn't look good on the SEM, and you like everything else to put it in the ultrasonic, but we killed several of those specimens, we broke several of those specimens by putting them in the ultrasonic well-meaningly to clean them up, but those little spines are so fragile, they just, you live and learn.
Ben, I have a question, if that's okay.
Yeah, go on,
It's,
Clive.
it's Clive here. So, hi Elizabeth, thanks for the talk. It's very interesting. I think you said in the talk that we're not sure or we don't know how the organism knows that its shell structure or the perostracum has been damaged, but it still manages to repair it.
Yes.
If that's the case, presumably there is some feedback loop. Goodness knows what it is, perhaps chemical or some other physiological form. But it's a really interesting one. Because it probably applies to more molluscs other than marine bivalves. So, any hunches on that, or do we have anybody who's looking into it? Do you know?
Well, it's one of those sort of stupid things that we all saw and didn't really think about. So once somebody sort of pointed out to you, can see it, and they very definitely thicken under the damaged bit, so it's very targeted. So they must be sensing it. Wonder slightly sometimes whether it's light. Somehow they can sense the light, and there's sort of ideas that some things like pinners actually are very good at sensing light on the outside of the mantle. I mean, that's just floating that out in the air, but it's very difficult. If the breach was total, then it would be easy to understand, but the breach is not total. There is still shell material between the mantle and the outside world. So they're sensing it somehow, but I don't know how.
Yeah, I guess the fun thing about this, it just illustrates the huge complexity that lies behind all these processes and how difficult they are, how cryptic they can be. But yeah, it's really interesting. Thank you.
I'd be interested to know if land snails did it actually. It's a job for you.
I have one comment about that. It's a general phenomenon that if you cut a land snail, if you section it, you find that the shell is a very consistent thickness, at least on the outside, right from the early whorls to the late whorls. But those whorls have had a totally different ontological history. The ones that near the apex have started off very thin and over a long period have been thickened from the inside, whereas the whorls towards the aperture have been formed thick almost from the start. And it always amazes me how the regulation ensures this. So it's the same point really about how they can respond to damage.
Yeah, that's interesting, yeah.
I have a question regarding the mantle cells. So is there any cell biological difference between mantle cells that lay down aragonite and those that lay down calcite?
We're sort of trying to begin to look at that at the moment. There's no immediately obvious one, but and it's not me that's doing this kind of thing, but people are beginning to see different genes switching on and off in different cells. So undoubtedly, yes. And it's very when, I mean, myself included, I've always produced pictures of the mantle with this big gap between it and the shell that's laid down. It's called the extrapaleo space, and we used to draw diagrams that were just this huge sloshy thing. Nowadays, when we're able to cut animals which are still alive, the gap between the mantle and the shell it's laying down is a matter of nanometers. So it's a very, very closely allied to the cells underneath. It's not all happening in this big cavernous sloshy pool called the extrapaleo fluid.
Would it be possible to, let's say, transplant a piece of mantle from a aragonite producing animal to a calcite producing one and see whether it changes?
I guess you could try. I haven't done it. I guess you could try it. I'm not very good at knowing about tissue splicing into things, but yeah.
Thank you.
Right, on that futuristic sounding note, then thank you very much. I would like to give Liz another round of applause, and then I'll stop with them.